Welcome to Sketchy. We take all the super complex stuff you need to learn and turn them into memorable visual stories packed full of everything you need to know on test day. Click the link in the corner or description to try for free for 7 days. Now let's get to it.
In this sketch, we'll walk through the 6 different classes of enzymes, which are proteins that accelerate chemical reactions. Let's start with oxidoreductases. This class of enzymes catalyzes oxidation-reduction reactions, which involve the transfer of electrons between molecules. That's why this bee is transferring minus sign pollen between flowers in a rusty oxidized pot with a reduced price. Alcohol dehydrogenase is one example of an oxidoreductase. It facilitates the transfer of an electron from ethanol to NAD+, which forms acetaldehyde and NADH. Well, I forgot my EpiPen, so let's move on from these bees. And speaking of moving, transferases are enzymes that transfer functional groups from one molecule to another, just like this gardener transferring a plant from one pot to another. For example, kinases are transferases that move phosphate functional groups. Pyruvate kinase is a super important one that transfers phosphate groups to ADP to produce ATP. Looks like the intern put the sprinkler on full blast and is splitting apart those flowers. Not best practice, but it is a convenient reminder that hydrolases are a class of enzymes that use water to break bonds in a molecule. For example, acetylcholinesterase uses water to break acetylcholine into acetic acid and choline. In the body, this ultimately stops actin-myosin interactions and allows muscles to relax after contraction. Our next class of enzymes are lyases. Hence, this gardener lying down to get the perfect angle for that chop. And like this gardener, lyases break bonds without water. Interestingly, lyases don't transfer electrons, which means they tend to make molecules form rings or double bonds, just like this flower that's doubly supported by its stem and a stick. And I hate to name drop, but you know Krebs? Like Krebs cycle Krebs? Well, that pathway includes a lyase called pyruvate decarboxylase, which breaks the pyruvate molecules from glycolysis into acetaldehyde, releasing some of that exhale-worthy CO2. Now, we're only representing bonds breaking here, but this guy probably rocks a sweet synth after work, because lyases that work in the opposite direction to form bonds without using water are called synthases. Next up, there's isomerases, which you can remember by the icicles on these tulips. Seems like the intern mixed up the fridge and the freezer again. Isomerases are a class of enzymes that create isomers, or rearrangements of a molecule that don't change its molecular formula. Sorta like how these are both tulips, they're just in different formations. And if you think giving someone tulips is sweet, try gifting them some glucosey candy, because glucose isomerase is one cloying enzyme. It rearranges the C6H12O6 in glucose into fructose. What better for your honey? And finally, our last class of enzymes are ligases. Ligases create bonds between similar molecules, kind of like how these similar flowers are being brought together with a ligature to make bouquets.